Thank you very much for the kind introduction. I'm Wei Lo from the Chinese University of Hong Kong, and is the project coordinator of the third one film-based research scheme project entitled the Systematic Development of Molecular Targets for Nasopharyngeal Carcinoma. This project is a joint effort of the research group from the University of Hong Kong, the Chinese University of Hong Kong, and Beijing Genome Institute. Our team member, including the expertise in the clinical science, genome biology, basic and translation research, and also the bioinformatics studies. In addition to our team member, we also collaborate with a number of the researchers from the local institute, for example, the Hong Kong Baptist University and the Hong Kong University of the Science, as well as the International Institute, for example, in Dana-Farber Cancer Institute and also the University of Toronto. On behalf of our team members, I will report the major finding of this 5-year NPC genome project. So actually, I will briefly introduce the background of the nasopharyngeal carcinoma. In short, we call NPC. NPC is a cancer arise in the nasopharynx region. It's behind the nasal cavity and in the upper part of the larynx region. And this the view under the endoscope. You'll find that there's a tumor mass here, and then while we cut it into the section and recognize the tumor cell under the microscope, we found that it distinct from other head and neck cancer. They are always with the poor differentiate or undifferentiated appearance. And most of the time, there's a heavy infiltration of the lymphocyte. And most of the distinct feature is that all of these cancer cells are infected with the Epstein-Barr virus. We call it EBV. NPC is a common cancer in South China as well as the Southeast Asia. Worldwide, there are there are about 130,000 new cases per year, and more than 75% of these cases are reported in this Asia area. So while we look at the incident, we found that the highest incident and all around the world is in the city in the Great Bay Area, including Hong Kong, for example, the others such as Zhuhai. And the cause of this cancer is mainly due to the EBV infection and also the genetic susceptibility to the Southern Chinese, especially the Cantonese. So it also called the Cantonese cancer. And it also relates with the consuming of some traditional diets, for example, the saltfish, and within this preserved food, there may be some carcinogen, like the nitrosamine, will damage the genome in our nasopharynx epithelium. So unlike other cancer, NPC always affect the younger age group. In Hong Kong, it became the first common lung cancer in the the younger male age at 20 to 44. So it become an important health problem in our community. The worst is that the patient, while they found that they have the NPC, they already have the advanced disease. While we look at the patient diagnosis, almost 17% of them are with the stage 3 and 4 disease, and the tumor are metastasis to the distant lymph node and also go to the other organ. For example, in this case, the metastasis were found in the lung. Currently, the standard treatment for NPC is the radiotherapy or the concurrent radiotherapy and chemotherapy. However, this treatment is not quite effective for the patient with the advanced disease, and they always develop the recurrent tumor. And unfortunately, until now, we did not have a effective treatment strategy for the patient with advanced disease and recurrent tumor, and we lack of the molecular target treatment. It's because there's a great knowledge gap on our NPC genome. Before we start this NPC genome project, we did not have too much information about the somatic change cause this cancer. So to address this problem, we have conducted a comprehensive NPC genome project to systematic discovery of the aims to systematic discovery the local molecular target and predictive marker for development the innovative effective treatment strategy to help this patient to prolong their survival and reduce the mortality. So we applied the advanced next generation sequencing technology to identify the genome landscape of a number of the NPC, and it allowed us to reveal the molecular mechanism and the key driver event contribute to the NPC development. Based on this knowledge, we can discover the biomarker and therapeutic target. And together, our team members have paid a great effort to establish and characterize the representative patient-derived NPC model. By using this model, we can based on this model, we can test whether this new treatment option or to develop the innovative therapy to provide the new treatment option to the NPC patient. So in this project, we have our clinical colleagues have collected about more than 200 NPC tumor. And we get this tumor either by the endoscope or by operation. And we, with the patient consent, we get this tumor specimen and then we cut it into the section. And under a microscope, we can precisely locate the tumor and then use the microdissection to isolate this tumor cell from the background normal stromal cell. And it allows us to obtain the higher purity tumor DNA for the next generation sequencing analysis. To the end of our project, we have generated 2 batches of the high quality NPC genome sequencing data set. And this and it made us to be the global, we have become the leader in the NPC genome research. So we have released the whole exon sequencing data set of more than 100 cases at 2017. And this year, we also deposit the final whole genome sequencing data set of 70 normal tumor pair into the Europe genome database. And this important genome resource now can be accessed by the scientists all around the world, and then allow them to use them in their research and promote the NPC study in different aspects. On 2017, we have launched the first version of our NPC genome map. In this whole exon sequencing study, we have firstly identified a number of the key driver mutations. This including multiple mutation of in the negative regulator of an important oncogenic pathway we call the NLKB pathway. And we have also done the functional assay to prove their driver role in NPC is important in the growth of this NPC tumor cell. And we have report the first time the involvement of a group of the gene involved related with the major histocompatibility complex, the MHC molecule. And this mutation will mainly target the host immune response. And We also demonstrate this NF-κB gene mutation contribute to activate the NF-κB signal in NPC. And then by the histological and pathology study, we also demonstrate this activate oncogenic signal occur in almost all of the NPC tumors. However, this mutation in the whole exon on sequencing study, we can only identify in about 20% of our case. So we believe that there are other genetic alterations involved in activating the NPC oncogenic pathway. So we have further to conduct a more comprehensive approach, the whole genome sequencing, to accurately detect the other genetic aberrancy, including the copy number variant, structural variant, and also the mutation in the non-coding regions. So by the whole genome sequencing, we can successfully identify the amplification of a number of the oncogene in the cell involved in the cell cycle, and we also proven that this NF-κB gene are also inactivated by the homozygous deletion and other type of the copy number variant. Furthermore, we also demonstrate the gene are also disturbed by the structural alteration in some of this tumor. The translocation will break up the gene and also lead to loss of the tumor suppressor property. So at the end of this tumor, we have completed the whole genome sequencing. At the end of this project, we have completed the whole genome sequencing of 70 NPC tumor. It allows us to make up a catalog of the somatic alteration and to define the key oncogenic event in NPC tumorigenesis. So based on this genome information, we will identify either somatic genetic alteration target the key oncogenic pathway. So in this figure, we demonstrate the incidence and type of the mutation on commonly involved in NPC tumorigenesis. And we found that the most affected mechanism is the cell cycle pathway and also the NF-κB pathway. The somatic traits also target the adaptive and also the innate immune response of the host. So although there's a less common mutation also found in other signaling pathway as well as the DNA repair mechanism indicate their potential role in the cancer progression. So based on this information, we can have a better understanding of the molecular mechanism for NPC tumorigenesis. And we made up a comprehensive model for the NPC development. It demonstrates the interplay between the EBV latent infection, the EBV gene, and also work with the mutation to cause the cancer initiation and progression. So among them, the most important mechanism is that we found that more than 60% of the cases are with the mutation in NF-κB pathway. And interestingly, in the case without this mutation, we found that they have expressed a viral oncogene called LMP1. And the LMP1 is well known to activate the NF-κB signal, or which have reported in our early study. So both of them can work together to drive the different hallmark of the cancer. For example, induce the cell proliferation, survival, and promote the inflammation. And one of the major contributions is that this NF-κB signal is that they will induce the release of the chemokine and the cytokine to recruit the T cell out to the tumor site to support the tumor growth. However, this while the tumor recruit the T lymphocyte into the tumor site, they also face the problem is that this T lymphocyte is an important part of the host immune response. And then they will, in the normal situation, they will kill by this cytotoxic T cell. However, we also looked that in the NPC genome, some of the tumor also acquired the somatic mutation disturb the MHC molecule, or they will express another EBV gene called the BNLF2A. Actually, the BNLF2A play an important role to inhibit the transport of the viral antigen or the neoantigen onto the MHC molecule. And the somatic mutation or in this molecule can further prevent present of this viral antigen to the surface of this cancer cell, and then it allows the tumor cell are not able to recognize by the T cell and escape and allow the tumor escape the host immune response. So recently, our clinical colleagues have performed the phase 2 clinical trial for using the PD-1 antibody as an immunotherapy in the metastasis and also the patient with the recurrent metastasis tumor. Although we found that 38% of the patients respond for this tumor, and we also found that most of the patients are resistant to this immunotherapy. Our new finding maybe suggest that this patient may harbor this loss of the MHC molecule, or they express the BNLF2A viral gene to escape the host immunity. So now we have conducted a phase 3 clinical trial for this immunotherapy, and we will using this new biomarker to identify the patient with the susceptible to the PD-L1 immune checkpoint inhibitor treatment. So our study have further identified a number of the molecular target can be treated by the FDA-approved drug. For example, the TACC FGFR mutation in about 4% of the patient, and the BRCA mutation in 5% of the patient. So we can we proposed this FDA drug to treat this patient, but the clinical benefit should be further examined in a large-scale basket clinical trial. So our team member, George, have also established a number of the representative EBV positive NPC model, including the cell line and xenograft. And then we, with the support of this film-based project, we have done a comprehensive characterization to demonstrate all of them are with the EBV expression, and we have also identify which EBV gene are expressed in this cell line, and we have also have a detailed genetic map for this tumor. So by using this model, we can able to test whether the NPC are susceptible to other FDA-approved drugs. So in our recent study, we have demonstrated the NPC tumor are quite sensitive to the FDA-approved sign in CDK4/6 inhibitor, and then it suggests that it becomes the less effective treatment option to the NPC patient. So on the other hand, we also demonstrate in 50% of our patients are with the chromosome 9 IP21 homozygous deletion, and it results in the loss of the gene called MTAP, which recently reported to be a synthetic lethal target of the MAT2A inhibitor. And this inhibitor already approved by the FDA for the phase 1 and 2 clinical trial. By the in vivo and in vivo study, we have also demonstrated the MTAP delete NPC are susceptible to this drug, and this encourage finding and promote us to contact our clinical oncology to see whether we can also use this new drug to run the clinical trial in this subgroup of the NPC patient. So finally, actually, the NF-κB is the major oncogenic target, but until now, there's a no good NF-κB inhibitor can be used in the cancer treatment. So our team member, Professor Alice Wong, have developed a novel gene therapy strategy, use the dendrimer with the siRNA, and then by this license, this patent technology, we are demonstrating have a quite effective to treat the tumor in vivo, in vitro, and in vivo. And we will further proceed the preclinical and clinical study, and hopefully we can apply this technology in NPC patients. So finally, I just summarize our achievement, and we have published a number of the high-impact journal paper in the journal, including the Nature Genetic, JCO, and Nature Communication. And we have also trained a number of the postgrad students, postdoc, and also the young faculty in the NPC research and also the genome study. So we also disseminate our information to the public. And finally, I would like to thank you again to the patient involved in this treatment. Without their collaboration, we did not able to complete our NPC genome project. Thank you very much for your attention.
Perhaps I could start actually by asking the first question.
Okay.
You showed actually just now very beautifully that a lot of these genomic aberrations that you have identified actually does translate to treatment. Now, we know that the primary tumor of NPC maybe is radiosensitive, but probably the problem is with metastatic NPC. So do you see any of those discoveries that you have made can actually be translatable to metastatic NPC therapy or potential sort of further developing it?
Actually, we have a number of the target identified from our NPC genome project. So in our presentation, I try to limit our report our finding on some the clonal genetic alteration. For example, the NF-κB alteration, and also the MTAP homozygous deletion. So it occurs not only in the primary tumor, and it also can find in all of these metastasis and also recurrent tumor, because most they are the key genetic changes are involved in the early development of the cancer. So it should be a promising and therapeutic target for the NPC treatment. So that's why I emphasize that we should address the focus on the NF-κB pathway in our presentation.
Okay, so you're recommending whether it is primary or metastatic, try to go and drug the major driver pathways, which is the clonal event, which probably would be more rewarding. Yeah. Actually, I agree with you, Ryan, for that one. Right, still no question coming in. So if I could also grab the second chance of asking the second question. So we all know that immune checkpoint inhibitors are extremely hot at the moment. So you just now showed that the checkpoint, the anti-PD-1 therapy actually is showing 30% response. Do you have any idea of the 70%? Why is it not perhaps sort of showing a response? Do you have any sort of insight into this?
So actually, I think in clinical study, we also have examining under PD-L1 expression. So most of the case, they are PD-L1 positive, but some of the exception case we cannot explain by the PD-L1 only. But according to our genome data, we found that some of the tumor, they have already have the MHC defect. So while it allowed the cancer cell did not present the neoantigen to the cytotoxic T cell. So even you resume the you inhibit the PD-L1 and then resume the cytotoxic T cell activity, they are not recognize the tumor. So that's why a number of the cases are not able to effective treatment by the immunotherapy. Another new finding is that we found that some of the tumor, they express at the high level of the viral protein called the BNLF2A. So they also target the antigen presentation mechanism. So the result, just like the loss of the MHC molecule, try to make the tumor cell escape from the T cell recognition. So the T cell, even they have restored their activity, they are not able to find their target.
Okay, so I have here actually 3 questions that just came in. So I'm going to go one by one. So the first question here is: do you think that immunotherapy drug can work synergistically with RT or any innovative radioimmunotherapy regime that can be explored? So your view on that?
Actually, I think that the radiotherapy can help for some of this case is because while you treatment while during the radiotherapy, you can induce more antigen and release into the environment. But the point is that according to our genome data and also the study of the viral gene expression, we believe that at least more than 50% of the case, they are not able to recognize by the T cell. So the T cell based immunotherapy may not be useful in this patient. So that's why we need to implement this two mechanism as a biomarker in future immunotherapy to identify which one should be benefit from the immunotherapy and which one are not. So for the case did not with this MHC mutation or the viral gene expression, I believe that we need to do more. For example, we need to develop the CAR T to directly recognize the cancer and not depending on the traditional antigen presentation mechanism.
Okay, so still in the subject of the immunotherapy, I have another question here for you. Is there any chance that gut microbiota can play a role to affect the response of NPC patients to immunotherapy?
Microbiota.
Yeah.
So actually, I think that I did not have any information on that. But I think the major drive I think the major driver in NPC already is the Epstein-Barr virus. Actually, the Epstein-Barr virus infection should be the major driver, but not the other microorganism.
Okay, thank you. So then I will go to the last questions, which is on using the multi-omics approach in therapy. So do you think there is a role for multi-omics to explore an optimum management therapy or strategy for those patients who are already suffering from recurrences or advanced disease? So sort of really how you see multi-omics genomics.
I think that our NPC Genome project is just a start point to enhance our to fill up the knowledge gap on the molecular mechanism of nasopharyngeal carcinoma. So we have NPC is well known is with the global hypermethylation in a number of the promoter. So and also we have only few information on the proteomic part of the nasopharyngeal carcinoma. So we still have a room to look at integrate the epigenome and also the proteome with the current genome database to identify more target and important biomarker for this cancer.